going to see the nitrogen fixation in non-legume plants. Ulnes and Casuarina are non-legume plants which contain the bacterium Frankia. Frankia will convert atmospheric nitrogen into nitrate, but the Frankia is associated with the Ulnes and Casuarina symbiotically. Psychotria is another non-legume plant which contains the bacterium Klebsiella. Klebsiella also a symbiotic bacterium, and that is symbiotically associated with Psychotria. This Klebsiella will convert atmospheric nitrogen into nitrate like that of the Rhizobium bacteria associated with legume plants, root nodules of legume plants. Now we are going to see the nitrogen fixation without nodulation. The following plants and prokaryotes are involved in nitrogen fixation. Number 1, lichens. Lichens is a double organism, it's a dual organism in which algae and fungi are mutually benefited, mutually related, symbiotically associated in lichens. Lichens contain Anabaena and nostoc. Anabaena and nostoc are the cyanobacteria, blue-green algae, which convert atmospheric nitrogen into nitrate. The second one is Anthoceros. Anthoceros is a bryophytic plant. In the thallus of Anthoceros, a cyanobacterium is living that is called nostoc. Nostoc is symbiotically associated with the thallus of Anthoceros. The Anthoceros is a bryophytic plant. The third one is Azolla. Azolla is a minute, it's a very small hydrophytic pteridophyte which contains Anabaena azollae. Anabaena azollae is symbiotically associated with the thallus of Azolla that also will convert atmospheric nitrogen into nitrate. The fourth one is Cycleg. Cycleg has coralloid root. Coralloid root is a special type of root, dichotomously branched, that is produced from the lateral root of the Cycleg. In the Cycleg coralloid root, there are two cyanobacteria are living. One is Anabaena and the second one is nostoc. Both Anabaena and nostoc will convert atmospheric nitrogen into nitrate. Now we are going to see the non-symbiotic nitrogen fixation. There are some free-living bacteria and fungi also fix atmospheric nitrogen. Number 1, aerobic bacteria. Some of the aerobic bacteria, free-living bacteria, will convert atmospheric nitrogen into nitrate. They are number 1, Azotobacter, number 2, Bigerenkia, and number 3, Detzia. The second one is anaerobic bacteria. Clostridium is an anaerobic bacteria living in the absence of oxygen environment that also will convert atmospheric nitrogen into nitrate. The third type, photosynthetic bacteria. There are some photosynthetic bacteria like Chlorobium and Rhodospirillum will convert atmospheric nitrogen into nitrate. Chemosynthetic bacteria, disulfovibrio Sulfovibrio is a chemosynthetic bacteria that will carry out photosynthesis by using energy obtained from the oxidation of organic and inorganic substances, and that is used for the preparation of carbohydrate during photosynthesis. That type of bacterium is a chemosynthetic bacterium. The disulfovibrio is a chemosynthetic bacteria that also have the capacity to convert atmospheric nitrogen into nitrate. Some of the free-living fungi also convert atmospheric nitrogen into nitrate, that is yeast and pleureria. Yeast and pleureria, these two are the free-living fungi which can convert atmospheric nitrogen into nitrate. Apart from that, some of the cyanobacteria also can fix the atmospheric nitrogen into nitrate. Number 1, nostoc. Number 2, Anabaena, and number 3, Oscillatoria. The nostoc, Anabaena, Oscillatoria, when they are in the free-living condition, they can also convert atmospheric nitrogen into nitrate. Now, we'll pass on to the nitrogen cycle and nitrogen metabolism. Nitrogen cycle consists of the following stages. Number 1, fixation of atmospheric nitrogen. Number 2, nitrification. Number 3, nitrate assimilation. Number 4, ammonification. Number 5, denitrification. First, fixation of atmospheric nitrogen. The nitrogen which is present in the atmosphere is a dinitrogen molecule, and that is reduced by addition of a pair of hydrogen atom. So the triple bond between the two nitrogen atoms, N, triple bond N, are cleaved to produce ammonia. Nitrogen fixation process requires nitrogenase enzyme complex. That is a very important enzyme complex. Nitrogen fixation process requires nitrogenase enzyme complex, number 1. Number 2, minerals like molybdenum, iron and sulfur. Anaerobic condition, ATP, electron and glucose 6-phosphate as H plus donor, hydrogen donor. Nitrogenase enzyme is active only in anaerobic condition. To create this anaerobic condition, a pigment called hemoglobin is synthesized in the nodules and acts as oxygen scavenger, and that will remove the oxygen. So that will create anaerobic condition. What is anaerobic condition? Without oxygen, the environment is called anaerobic condition. Nitrogen fixation bacteria in root nodules appear pinkish color due to the presence of leg hemoglobin pigment. Overall reaction for this process, N2 plus 8 electron minus plus 8 H plus 16 ATP, that gives 2 NH3 plus H2 plus 16 ADP plus 16 inorganic phosphate. So that is the overall equation. The second step is nitrification. ammonia NH3 plus is converted into nitrite NO2 minus by Nitrosomonas bacterium. So nitrate is then converted into nitrate NO3 minus by Nitrobacter bacterium. Nitrification is a process of conversion of ammonia into nitrate in two steps. In the first step, ammonia is converted into nitrite by Nitrosomonas bacterium. In the second step, nitrite is converted into nitrate by Nitrobacter bacterium. Plants are more adapted to absorb nitrate NO3 minus than ammonium ions from the soil. So that can be represented as 2H3 plus 3O2 in the presence of Nitrosomonas, that is converted into 2NO2 minus plus 2H plus 2H2O. The second step, the 2NO2 minus, that is nitrite, combined with oxygen and form 2NO3 minus nitrate in the presence of Nitrobacter bacterium. So that is the reaction taking place during nitrification. The third step in the nitrogen cycle is nitrate assimilation. The process by which nitrate is reduced to ammonia is called nitrate assimilation and occurs during nitrogen cycle. This is the equation given under this nitrate assimilation. NO3 minus, that is the nitrate, is converted into NO2 minus by nitrate reductase enzyme in the presence of molybdenum. NO2 minus is converted into NH3 plus by the enzyme nitrite reductase in the presence of Cu plus Fe. So that is the reaction taking place during nitrate assimilation. The fourth step is ammonification. Decomposition of organic nitrogen, that is the protein and amino acid from dead plants and animals into ammonia, is called ammonification. Organisms involved in this ammonification are Bacillus ramosus and Bacillus vulgaris. So what is ammonification? Ammonification is the formation of ammonia from the dead plants and animals. That process is called ammonification. The dead bodies of the plants and animals contain organic nitrogen, that is the protein and amino acid, that will be converted into ammonia by two bacteria, Bacillus ramosus and Bacillus vulgaris. That process is called ammonification. Denitrification. The next step is denitrification. Nitrates in the soil are converted back into atmospheric nitrogen by a process called denitrification. Bacteria involved in this process are Pseudomonas thiobacillus and Bacillus subtilis. And here also the reaction is given. This is the overall reaction for this denitrification. Nitrate NO3 minus is converted into molecular nitrogen by Pseudomonas bacteria. Now we are going to see about nitrogen metabolism or ammonium assimilation or fat of ammonia. Ammonia is converted into amino acids by the following three processes. Number one, reductive amination. Number two, transamination. Number three, catalytic amination. First one, a reductive amination. Glutamic acid or glutamate is formed by the reaction of ammonia with alpha-ketogluteric acid. During this process, this is the reaction formed. Alpha-ketogluteric acid plus NH4 that gives rise to glutamate plus H2O. By the action of glutamate dehydrogenase enzyme GDP. During this process, NADPH plus H plus is converted into NADP. NADP means nicotinamide adenine dinucleotide phosphate. The second process is transamination. The transfer of amino group, that is NH3 plus, from glutamic acid or glutamate to keto group of keto acid. Glutamic acid is the main amino acid from which other amino acids are synthesized by transamination. Transamination requires the enzyme transaminase and coenzyme pyridoxal phosphate, that is the derivative of vitamin B6 pyridoxine. These are all the process reaction taking place during this transamination. Amino acid 1 combine with keto acid 2 and form keto acid 1 and amino acid 2. Amino acid 1 R1 CH NH3 plus COO minus. Keto acid 2 R2 C double bond O COO minus is equal to this combined with this and form the keto acid 1 R1 C double bond O COO minus plus R2 CH NH3 plus COO minus, that is the amino acid 2. Third process is catalytic amination. The catalytic amination is always called GS pathway or GOGAT pathway. During this process, during catalytic amination, glutamate amino acid combines with ammonia to form the amide glutamine. The glutamine reacts with alpha-ketogluteric acid to form two molecules of glutamate. That is given like that: glutamine plus alpha-ketogluteric acid, that is 2-oxoglutarate, that gives rise to 2-glutarate in the presence of the enzyme GOGAT enzyme. During this process, NADH plus H plus is converted into NAD. So that's all the reductive amination, transamination and catalytic amination. By these three processes, ammonia is converted into amino acids. That's all today's topic.